Hello, friends. Welcome to another episode of the EverymanBio podcast. My name is Josh McGinnis. I want to start off today just by wishing you a happy fall, happy autumn. This is one of my favorite times of year. Thanksgiving is by far my favorite holiday, but also for other fellow mushroom hunters out there, this is a special time of year. Tends to be quite fruitful for those of us who like to go out and forage, looking for fungal specimens and new species that we maybe have never discovered before. This week we have a very special guest by the name of Craig Trester. Craig is a mycologist located in the New York City area. I found him through Instagram, and also he helped mentor Secret Jacob in learning DNA barcoding, who then has now mentored me in learning DNA barcoding of fungi as well. Craig is one of those people that's incredibly knowledgeable. He has a deep technical understanding of fungi, all the way from the genetic level to the role that it plays in our ecosystem, and how we can use fungi to build sustainable practices in our society and in the world, the impact that it has on soil ecology, and just the general environmental impact overall. And I could have probably talked to this guy for many hours, like so many of my other guests, but we covered a lot of ground and a lot of really interesting information. You can tell that he's really honed in on his ability to teach. That's something that he spends quite a bit of time doing these days, is teaching other people who are new to mushroom growing or new to learning about mushrooms and their role in the environment. And you can really sense that come through with just the way that he goes about sharing the information and teaching us all of the knowledge that he has. So I think you're really going to enjoy this episode. So without further ado, Craig Trester, ladies and gentlemen. Thankful to have you as a guest. And
Yeah.
our circle of people we keep intersecting with different people in our groups, which is really cool. So Sigrid's told me you were a big part of helping mentor her with learning DNA barcoding.
Yeah.
I know we both know Damon Ty now as well, who's
Yeah.
amazing. He was on the podcast last time. Uh, Perhaps you could just start by sharing us a little bit about like yourself, what you do. Your Instagram says you're a mycologist. Maybe
Yeah.
what does that mean for people who don't even know
That
what that word means?
so yeah, that's a great question, and I think it's a great way to start off the session. So to clarify in general, so mycology is a pretty young science as a formal as it was only in 1969 that fungi were formally separated from plants. They were given their own dispensation as their own taxon. So it's pretty interesting because whereas in the study of plants, so you have botany, but also for the science of growing plants, you have horticulture and the science of managing the production of crops, agronomy. You have all these different subdisciplines. But when it comes with mycology, because for the longest time that people thought they were like these kind of very old non-photosynthesizing plants that like reproduce by spores, like kind of like liverworts or ferns. So they kind of fit in, but so it's really interesting because, with mycology, there are people who, go to go through academia, go through their master's and their PhD programs, either studying animal or plant pathology, which is, pretty prominent. It's understanding because, fungi are prolific in our world, and they're basically their metabolisms are serving the capacity to cycle nutrients, both living, non-living, and then even mineral in this world. So in human beings, agriculture, growing things, or even just biotechnology, we tend to forget that that the plow, the stick that was the first thing to push a furrow to allow us to domesticate plants, or even a yoke that went around oxen that we domesticated, is just as much a piece of biotechnology as a pipette. We tend to forget that. And so that's the whole thing about it. So, our attempt to work with nature, to get it to function the way we want it some of the time, is that attempt. So understanding the whole cast of characters that interact with us and biology in a kind of in an anthropogenic way through agriculture, but also in the natural kind of cycles as well. So yeah, so back to the point with mycology, so either so with plant sciences, there's botanists, there's plant genetics, there's horticulturists, agronomists, but with mycology, it's kind of all one envelope because it's so new. So a person who studies it, it's something, it's a title of respect that I've even thought to myself, maybe I'm starting to call myself an applied mycologist. because my background is very much through the DIY aspect, but my entry point into mycology was through kind of environmental science, looking for bioremediation, understanding the roles that fungi play, their natural metabolic capacity, how we could, we could interact with them in arrangements that simulate the natural roles to, to utilize some of their abilities, not only to decompose, but also recompose in certain ways. So it's something where, as I've gotten more along in my path, like I definitely want to pay gratitude to the people that have gone to school for it and spent the better part of a decade getting their PhD to be called a doctor behind it. But there's so many different dispensations, but we're entering a point now where kind of the very interesting DIY nature of mycology, through the whole capacity of how, the McKennas did kind of underground cultivation, but that led to people basically, who learned how to transfer those skills. People that maybe started in a clandestine kind of way, cultivating select compost-loving species, realized that these were transferable skills to
I'm one
grow
of those
a variety.
people, by the way.
Yeah, it's something interesting because it's the factors like, oftentimes like most people, when they talk about, like fungi play a huge role in our ecology, but also in interacting with us, the fact that we can interact with them as a food source and recycling nutrients. And it's important we talk about all the variety of roles within the perspective of mycology, because most people's interaction with mushrooms kind of either ends in the kitchen cutting board of the music festival. But it's so much broader than that. That's a little joke I tell whenever I teach a class, because it's important we talk about these things that were taboo, but in a mature light, in a fashion that they have a huge amount of benefits. So it's kind of interesting where I've started in the environmental focus of mycology, but I can talk about the more kind of, cultural common associations, but also elevate them to like the deeper influence they play in our larger ecology. So I would call myself an applied mycologist. And one thing I've really gotten into now is understanding fungi with the environmental focus, the roles in generating, creating soil, and then even going further, which, Sigrid and Damon talk about, some of the molecular mycology. The fact that really the ways they interact and modify and express genes, interact with their environment on a molecular basis, both internally and both externally, with exudates externally and then internally with their unique multinucleation, cytoplasmic streaming, and gene expression, which is mind-boggling how they're able to do so much. Really, that's the frontier, because that can unlock so much more for the application of biotechnology or mycobiotechnology. So I guess in a nutshell, albeit a rather large one, that's what I consider myself to be. But yeah.
I mean, I have a similar experience with that. I started learning about mushrooms, just natural curiosity of like, what's this weird thing that grows out of the ground, and looking it up.
Yeah.
And then I found the clandestine community, and just like the mycelium, that sort of spread out into other domains, into fungi. And I just became enthralled and fascinated by this organism, which plays such a huge role in both the evolution of man, but also our world, and pretty much everything. That we actually, humans share more DNA in common with fungi than we do with plants. That's pretty amazing. I think most people don't know. And on that topic, what do you think are some of the misconceptions that the general public has about the world of fungi? And maybe you could speak a little bit about that, because I think I have a pretty straight and narrow family who don't understand or get why I'm like an addict to this stuff and going all in on it. And I think they have a cursory view of what it means to be into fungi, and it's related to drugs and psychedelics and things that culture has sort of built around the imaging.
Yeah, that's a great question. So it's just something very interesting, because like you either have a mycophilia or a mycophobia, oftentimes based upon the cultural background. So we're coming from a culture that is an English-speaking background, and so what's really interesting is that you look at a lot of the terms for fungi, they're referred to like toadstools or fairy rings. So these were things that were either associated with like, or even too, just like decomposition. A large number of herbalism that started in England. One of the head leaders of the herbalist, or founding leaders of herbalism way back when, really told people to avoid mushrooms, and so it was more focused on plants. You had this more focus to plants as opposed to fungi.
Mm.
Whereas if you go to continental Europe or you go to Asia or even Africa and even South America, and even in the Americas too. We understand that indigenous peoples in North and Central and South America, indigenous people all over the world have a very tight relationship with fungi, but it's more in the English-speaking culture that got a dispelling kind of bad rap. And there's even a theory I've heard of why in the English Isles more specifically was because they were an island. It was far harder to invade the island. So where if you were on continental Europe or other areas of disturbance where there were normal agricultural production, going into the forest and foraging your food, this high nutritious protein source and this lineage that's there was far more common in continental Europe and Eastern Europe versus the English speaking language. So it's funny that gets carried over into our cultural history. And then also too, kind of like the kind of fear of decomposition, the fact that they're ephemeral. We see,
associated with death is that where you're going?
Yeah, but they're all, but it's more so that they're mysterious.
I see.
So the factor is we see plants and animals far more often, whereas fungi, they pop up, they show up for a little bit, they decompose, and so it's kind of this mystery. And the fact they're there in the site of decomposition is something that kind of does confront our mortality. And depending upon your cultural background, mortality is a process of life or is something to be feared, and more so in kind of an intermediary point to a form of an afterlife. So it's these different connotations, but it's exciting to hear that we're kind of moving out of a fungophobia to a fungophilia. I think a number of things. One is, the prospect of people now, they're considering, protein sources that don't involve animals. So either eliminating or reducing their protein consumption from animals, and still getting a significant amount of not only protein, but nutrients and beneficial compounds in the actual fruiting bodies. The focus of medicinal fungi, which have a whole range of immunomodulatory compounds that can interact with these innate biological processes in our immune system that actually resemble the signals of fungal pathogen in the past. It's pretty crazy. So which stimulate this adaptive and innate this adaptive immune response from moving it from inflammation to actual cytotoxicity. So rather than inflammation occurring, it'll actually target cells that are either moribund or overinflamed, that can cause this other kind of mess of signaling. And even further too, what's more exciting now, which was kind of gained attention in the middle of the 20th century, but is now through an information embargo, through the war on drugs, we're actually realizing now the huge benefits for mental health, and
Absolutely.
also dealing with substance abuse of the psychoactive fungi. And the fact there are even results that are showing you're having benefits in upwards of 60 to 70% of the test population, which is unheard of in clinical settings. So it's something remarkable that we're learning. There's so much to learn about mycology, and that we've barely even kind of scratched the meniscus of what's there. So like, it's wide open. Like, and it's very hard to like focus and like stay in one thing, which I guess for myself is like when I was younger, it's hard to sit. I think a lot of them are current educational frameworks. A lot of students who may be brilliant in these very multifaceted things or approaches or fields struggle with this very narrow focused track. But when you kind of look to mycology, it's so appealing because it's so wide open of so many things you could do and explore, and then also encourage you different moments. So that's kind of that kind of adds on to the first question as well. So it's exciting that there are so many different applications to them. And a lot of the stuff that I do is for how they can recycle our waste streams preemptively. And I can talk more about the work I've done as like working with a high school student, mentoring on micromediation, but not after the fact of a brownfield or a spill site, but preemptively dealing with waste streams of paper that are often, with the Amazon effect, when you take cardboard or paper products out to recycle, unless your municipality has a dedicated system, most oftentimes they're going to a landfill. And however, we can kind of take that energy source, that carbon source that we've chopped down those trees and mass room turned them into pulp. We've done all the hard work for the fungi that actually evolved to decompose these structures, and turn that into something that can build soil or be an incredible amendment for agriculture. So that's kind of the application as well, where my focus is more on the environment. But yeah, we're kind of at this watershed moment with mycology, and it's another note kind of with your own journey with EverymanBio. I've definitely kind of been watching your journey as you started up, shared about your project and the diyBio on mushroom sequencing, and even back in kind of thinking back to 2017 when I was finding the community biology lab like spaces in the United States. New York City, and how I was cutting my teeth and having a dedicated lab space to work with fungi on agar and grow them out and experiment that, was in my kitchen. Oftentimes I'd have roommates, I'd be like my pressure cooker pouring agar, and have my petri dishes in the fridge, and my roommates would throw it out saying, "Why are you growing mold on like petri?" I'm like, "Well, it's not, but it is." But at the same time, that was with my own experience of learning molecular biology. So one of the things with the lab was to take some courses on it, but any organism you brought in that couldn't be identified from, let's say, like a curated culture bank, or like an organism bank, like American Tissue and ATCC, or I always mess up the acronym, but a certified culture preserving organization, you had to identify. So for me to even to work with the organisms with my micromediation project I was trying to do there, I had to barcode them. So
Oh, okay.
so flashing back, I look to myself when, and I love the ambition that you do. Like it's hard, it's something, but it's the persistence of that can do it in like little details. And I think back to when I would like go into the lab, like back when I was working a service job and a flexible, I'd run a reaction. I'd get there at 6, run the reaction. Maybe it messed up. Turns out at 8, 9 o'clock, I'd run another reaction. Like you take a nap on the couch. You'd read something on the couch, take a nap, wake up. It'd be like 4 a.m, finish it, run a gel, and like, oh, great. And then like put your samples, and then like go to my job and be amped on this, despite the fact that you just sound like a manic. Like, are you crazy? Like, yeah, maybe a little bit, because I'm so enthused into this. But yeah, it's exciting because mycology came into light formally because of molecular biology.
Mm.
We found out they're actually their own taxa, and even closer related to where that taxa sits in relation to eukaryotes because of molecular biology, because we can look at the regions of our genome that are more conserved or variable with relation to animals or plants, or other higher organisms than multicellular organisms. So it's kind of this amazing thing where in the 60s, 1969, they were formally separated as their own taxa, but it was in the late 70s, 80s, 90s when sequencing technologies became more and more affordable and more practical, that we could actually formally say through science, which is all about reducing uncertainty,
Mm.
that this is our current understanding of the situation. So yeah, it's kind of this interesting thing that like molecular biology and mycology formally are growing together in disciplines to define and refine one another.
Yeah, that's fascinating. Yeah, to touch on the barcoding aspect of it, I have some samples at the lab that hopefully should be getting sequenced and returned to me today. So I'm a little bit anxious. It's my second go around. The first time I got a bunch of junk data, but I learned a ton from that process. That's par for the course when you're when you're a DIYer trying to learn this stuff by watching YouTube videos and
Exactly.
reading
And
papers.
well, that's the thing about it. Whereas I think a lot of it is we tend to have a culture where we're afraid of failure, right? We're taught to avoid failure, right? We're taught to like do it right the first time. Like, and even people that are high performers, they'll all have this fear of like, if you're, let's say like hypothetically someone in high school and college, and I even see this in some students I actually teach and tutor and mentor, that let's say they're a straight A 4.0 student, the minute they get a B minus, it's like the end of the world, right?
I
It's
was
like,
that kid.
yeah. And but this is reinforced a cultural dynamic. It's like a fixed mindset versus a growth mindset. That
Well,
every
that's
little bump along your journey is something that you learn from and to improve upon.
You touched on something that's really personal and important to me, because that's why I started EverymanBio. I had reached a sort of peak of my technical career, holding the highest levels you can hold, and kind of reached all these places that I thought were going to be the place that the things that made me happy, holding certain titles, making so much money, da 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 da. And I realized that wasn't working, and EverymanBio was sort of, you know what, I don't, this is a completely new field. I wanted to learn synthetic biology. Actually, that's part of the journey. The DNA barcoding is a pit stop along the way to engineering an organism. So that's the next stage of EverymanBio, and something I'll be starting with pretty soon. But part of that was, you know what, I'm just going to put it out there. I'm going to put all the failures, all of the bumps along the road, all of the trials and tribulations. I'm just going to be honest and put it out there, because it's real. It's honest. Science is hard. It's even harder when you're doing it in your, dining room or wherever, because there's tons of people doing it on their kitchen tables. And so, yeah, I definitely understand what you're talking about there. On the topic of using fungi to help the environment and sustainability, I know that there's a fungi that can eat plastic, and obviously there's a lot of fungi that eats dead wood and dead material. But I've yet to see any real commercial breakthroughs other than I think there's a MycoWorks. It makes clothing from Ganoderma or reishi. I think it's the same thing. And then another company that makes mushroom packaging. But I'm still waiting for the time we can take our plastic bottles and they get dumped off at the city bioreactor where they just get tossed into a big fermenter and the mushroom just goes to town with, breaking it down. Why, what do you think is the reason for that? Or are we still too early? Is there more research to be done to get to that phase? Where are we in that? What do we need to do to get to that breakthrough moment where we, something like our recycling in Los Angeles doesn't actually get recycled. It gets, even though we have a separate bin for it, everybody knows it just goes right to the landfill.
It gets recycled.
Right.
Like, yeah, no, and this is something too, is that like, and it's really funny that like, especially in the 1980s and 90s, the whole thing of the 3 Rs: reduce, reuse, recycle. Even though the first one is like the reduce is the most important thing, right? Because they lead into like reusing and recycling. But it's kind of the whole thing. So getting into kind of the plastics, right? Definitely it's something that, so you'll often hear like Pedalopsilus microspora, mushroom found in the Amazon, degrades plastic. Well, it's polyurethane. And then, or Aspergillus niger, found in a garbage dump in Pakistan. Like Aspergillus niger tubiformis, which is actually the same like fungi. The Ascomycete grows like as black little specks on your garlic as well. But like this one type can like break down polyurethane, and it's something that, there is hope. And the factor is, like these organisms evolved to deal with a variety of like substrates, their biomass and food sources. You look at through all the extinction events, really the species of fungi, the species of organisms that really did not really experience a reduction in their biodiversity was fungi, which is kind of terrifying. You look at the big extinction event 6 million years ago, and they even found this out that like whenever there's a big extinction event, like they just team up with algae and become lichens for a while and just like just decompose all the organic matter. They're just like, ooh, dinner time. But yeah, like so now that we're in the Anthropocene, now that we're the geological epoch where human beings have gotten so good at making things that don't decompose, they becoming literally parts of the fossil record. It's something interesting to think about. So oftentimes this is also too that, fungi are new and they're exciting, but we tend to reduce them. Like it's kind of this part of the Cartesian reductionism that does happen that we're starting to realize. But the factor is that fungi, they're a part of a subset, they are the architects of the soil, but they interact with so many different organisms. So one enzyme that's really been really promising and looking more into synthetic biology is PETase. So PET is the plastic polyethylene terephthalate, PET. That's the reason why they give them acronyms, because like after a certain point it becomes an alphabet soup of consonants and vowels. But yeah, so this is an enzyme that's been found in certain types of bacteria that can decompose it. So the reality we're realizing now that as much as we talk about synthetic biology and genetic modification, we're realizing that horizontal gene transfer happens, not just between bacteria and the environment, but actually between fungi and bacteria.
Cross species.
Continually. And what's even crazier is, cross taxa and even cross domains, like from prokaryotes to eukaryotes, which is kind of crazy.
Incredible.
And then what's even further is that we're realizing that a lot of these fungi in the soil, they act as the architects of the soil that will actually make these structures that bacteria can travel along. There's an amazing video I can send you where you can actually see this Cunninghamium elegans, this one type of fungus, where there's thin films of water that Pseudomonas bacteria or Pseudomonas pudia bacteria, which are some of the more remediative ones that can deal with like tougher contaminants, that are running along this film of water on the surface of the mycelium. So it's almost like a highway. So what's even crazier is that they've realized that this can actually be a means for gene transfer. So if you have a contaminated site where there's like petroleum hydrocarbons or plastics or things that are leaking in the environment, you could have bacteria that are living in this area, the populations that are surviving, it's like, when you see like on Lysol or whatever disinfectant kills 99.9%, well, that 0.1% has some amount of genetic variance that helps it survive in this hostile environment. So the same thing goes for where we prospected these enzymes from microorganisms. So on this one cluster of, this is the hypothesis in general, is that this one cluster of contaminated area, you could have these genes being transferred to other populations around the same site, because you have this genetic diversity, this genetic specialty being treated to this contaminated site, and then along these filaments of hyphae, where there's a film of water where these bacteria are transferring, that's a vector by which you can exchange these different genetic diversity amongst populations of these more mediated bacteria. So getting back to it, so when we hear about a fungus that, like a mushroom or a fungus that breaks down plastic, like, well, it's more about them interacting with the whole trillions of microorganisms in the soil. And that's the thing about it. So this is something when I started with mycoremediation, definitely, Paul Stamets, like talking about his work breaking down petroleum, Peter McCoy, growing mycelium on cigarette butts. And that's all amazing, but it's still the reductionism. It's still like focusing on one type of species. When in reality, where do you find fungi in nature? They're breaking down trees as primary decomposers. They're decomposing older organic matter as secondary decomposers. They're breaking down organic matter turned into soil, compost loving species. And even further, a lot of the soil fungi, the Ascomycota, which are the majority of the fungi, breaking down what's left, continually like nutrients.
So are you suggesting we need to be looking at this problem as more of a system as opposed to these isolated components, which are probably easier to study and easier to break down,
but harder to arrange.
It's really the orchestration and the balance of all of these organisms working in harmony that creates the overall effect. So
it's the emergence effect that occurs. And so what's interesting is that given the conditions, and this is something I've really went down the rabbit hole on, because I learned more about from fungi. I learned more about regenerative agriculture, traditional ecological knowledge, permaculture, but also soil, how important soil was. And that, the role fungi play in generating soil, but also being the architects of soil that facilitate all this other biological diversity and facilitates interactions. And that the more biodiversity you have, the more niches of roles of whether food sources that are excrements or waste products will be utilized. In much like the way that waste products that we produce, there will be genetic diversity, because these organisms have very short lifespans. They're evolving at a much faster rate in dealing with their food sources and areas. So that's the whole thing about it, like where, potentially, could you look at a type of fungus and maybe, engineer like yeast with a PETase gene from like with a plasmid from another organism, like this cassette, and design it to excrete it and break it down? Yeah, you could, but the reality is, okay, what's happening after that fact, right? When you're constraining this one type of enzyme as the one answer, what's happening to that plastic? Is it degrading down into forms that are more stable, that are far more reactive, maybe even more toxic than the original versions? The same thing goes for petroleum hydrocarbons. And even two considerations that, this enzyme that's functioning, there might be another interaction with another organism that's actually making an enzyme that's allowing this reaction to exist between these two different enzymes. So it's like you get this emergent effect, right? It's the emergence of activity that is almost indescribable to happen. And the one thing that a number of people that have looked into mediating, riparian or aquatic ecosystems, John Todd and by which you have all these niches of biodiversity, but filtered them through different points by gradually increasing the biodiversity in disturbed ecology, which allows it to adapt to those shocks that normally would destroy like a native or an untouched or pristine ecology that would be disturbed. Or so the reality is, yes, that is a function that can happen, but it happens in interaction with the whole coterie, the whole entourage, the whole cast of microorganisms in those ecologies, in diverse ecologies.
That's interesting. When you say emergent, that immediately makes me think about the brain, which is something that I've been studying quite a bit in the last decade through my personal journey in therapy and interpersonal neurobiology, and very much consciousness and how the neurons work in networks and can communicate with each other creates these emergent experiences that we experience as our consciousness. But what's fascinating is when you look at fungi and you look at the networks and even the networks with other organisms, there's so much similarity between the brain and the brain's underlying structure and mycelium and the various networks that are created in fungi. That to me is incredibly fascinating. It's like life imitating art, art imitating life.
Yeah, it's miraculous, and it's something that we tend to think about. Well, we tend to anthropomorphize things, right? Like look at an electric socket, right? We're a very idiomatic species, but rightfully so. It's part of our culture, right? It's the fact that like you can look at something and see a human face in it. That's pretty important from an evolutionary standpoint of like having a socially organized species that can like the super brain thing. Like we're smarter together, working together as teams, rather than individuals, and in most scenarios. But getting back to point, it's kind of, you look at like a mycelial network, you'll look at these branching filaments that are present in, wiring systems, an anthropogenic concept, or human-made something artificial. But even further, look at our circulatory system, look at our respiratory system, look at our nervous system, look at the
The
dendritic
veins in a
trees.
leaf. Yeah.
Yeah, like veins. Look at the dendritic trees in our alveoli. And the reality in general is that when we look at us as organisms, right, we tend to think of ourselves as like a singular entity, but we're an assemblage of trillions of cells that are different, have all the same genome, that are expressing different parts of that genome. And it's pretty amazing, because that's the progression to where we got to more complexity. But still, in that complexity, you have those very simple ways of filling physical space, optimizing the way you basically can move resource materials through your body. Whereas with fungi, they're so simple, but they're manifesting all those roles into one kind of filamentous growth structure of their hyphal network that composes mycelium. So it's amazing, because you look at a mycelial, look at a hyphae, you look at the anatomy. They have an outer layer that is manoprotein, so sugar and protein kind of pellicle layer. You have these huge molecular cross-linked polysaccharides, these alpha and beta glucans that build the cell wall. You have chitin, which is the polymer, heat-resistant polymer, which makes them durable and tough. And then you have the phospholipid bilayer. And then pregnant this bilayer, you have receptors for heat, for chemicals, for light. It's kind of amazing, because all of these individual receptor proteins that are, they're pretty much bound to our kind of lipid bilayer in each of our cell types, are present at different parts of this mycelium. So it's such a simple organism, but it has such complexity in such a elegantly efficient form. So it's kind of mind-boggling, because but then you zoom out, you look at like a littoral flow of a river delta. You look at you even look at the kind of like these fractal filaments of these
I was going to say the Mandelbrot
lightning
set.
strikes. Yeah, and so it's something that there's this pattern in nature of how even on a mathematical level where people try to understand it. When exponents is kind of like psychedelics for mathematics, because you blow it up to this kind of aspect as well. But these elegant kind of iterations of patterns that optimize for kind of a space solving problem, that are almost kind of this ineffable standpoint you see everywhere in nature. It's kind of like this kind of goes back to classical philosophy, and even kind of people alchemy, like the macrocosm, microcosm, like the as above is below, and then in physics we know it is like a scale invariance,
which I'm very much fascinated by as well. And I think what's interesting is when you have a psychedelic experience, it reveals that interconnectedness that we have with nature and ourselves. And you could see more of those things more closely, which I also think is quite fascinating.
Yeah, it's interesting how, albeit Rachel Carson writing Silent Spring, Maury Bookchin writing Our Synthetic Environment, which are kind of like the literary ties to the environmental movement, and understanding that, the same time that these compounds derived from fungi, these metabolites from fungi, which interacted with our serotonin, our serotonin system, definitely kind of very abrupt way to deconstruct the anthrocentric narrative, that you're part of something much else going on. But yeah, it's a kind of interesting because there's definitely like the stoned ape theory about talking about potentially this might have been a way to increase our brain development. But it's an interesting thing because this cluster of genes that for psilocybin synthesis um, you know tryptamine psilocybin synthesis originated 20 million years ago. You know, roughly, you know, roughly we think about 13 million years prior than that our most distant simian ancestors came about.
Mm.
And so it's really interesting. A lot of people think, okay, what is this? What is this function does it serve? Some people think it's an anti-feed agent that we understand in times where there was far more oxygen on planet Earth. And we had arthropods that were like, you had millipedes that were like a meter long. Like, so you have,
Yeah.
we understand these organisms, and so we had this period of time with the megafauna, right? We're sitting on the ecological rubble of these distant, like, these geological epochs in time. Where, think about the megafauna. What about the megafunga, right? Like, think about these crazy roles, and that a good way to prevent an organism from eating your fruiting body that contained your genetic, your genetic lineage would be maybe to have some kind of system that was an endocrine disruptor.
Mm.
However, in an arthropod, they have a rather somewhat simple neurochemistry, neurobiology. But in simians and lower and higher apes, we have a little more intricate neurobiology. So this thing about it, like, that creates this unique context because we're a social species in that aspect. In addition to, I'm not sure, have you read Merlin Sheldrake's book, Entangled Life?
No, I haven't.
So he does a great job. He really goes down the rabbit hole because he did study mycorrhizal fungi. I think at the Smithsonian Research Institution down in Panama. But he talks about, we talk about the stoned ape hypothesis, but we also think about but also the drunken monkey hypothesis that that the gene that's responsible for us to turning ethanol into acetaldehyde, mutated and becoming 20 times more efficient to the point where ethanol wasn't toxic to us. Where when we actually might have evolved to basically eat fruit that was overripe because of yeast. So yeast played a role into it as well. And even further, the fact that we caught fire, we learned how to cook our foods, where far more real estate, like far more space that was normally reserved in our jaw muscles for chewing foods that were tough and raw was actually might have freed up more space in our to actually help grow this brain that might have been facilitated by the social interactions from fermentation, consuming things that might have maybe get you slightly intoxicated and, potentially. So yeah, it's pretty amazing kind of trying to do this prehistoric anthropology as anyone's guess, but a lot of the concepts, understanding that kind of, that fermentation, the aspect of like nutrition, like even with the aspect not of alcohol, but fermented foods playing a huge role in our own gut health and nutrition. And then, how these are socially, these are occurrences in our human history, the cultivation of us as modern humans that led us to this point of becoming a species that was far more social, understood cultured foods, and then potentially more so understood deeper meanings and complexity into the natural world of our own relations. So,
yeah, that's incredible. You do quite a bit of teaching, as I understand it. Where do you point someone who's brand new and wants to learn about mushrooms and soil ecology? Where do you start with them?
Yeah, so it's a great question, and the reality is there's different paths, right? It depends on who the person is, what their background, what their interest is. And this is one thing I always try to do with my students. There are many different kind of pedagogies or teaching styles or methods that, everyone's a different kind of learner. So one thing I've always found really important in the sciences, especially because there is a fair bit of things to get through and learn, to experience, to comprehend something that's fascinating, is to kind of create like a vantage point in the horizon they can understand. But answering the more simple question about like getting into where to start, learning generally about mycology and fungi. I think one of the book that really got me into it was Radical Mycology, written by Peter McCoy. 700 pages. It's a tome that covers, the history of fungi, ethnomycology, anthropomycology, cultivation, DIY science and remediation, recipes. It's pretty comprehensive. And that's something to the point where like it shows you the whole variety of directions you can go. and allows someone to kind of choose their own journey based upon their interest. Because my background, I tend to have more of a slant towards, environmental science and, like on the ecological aspect of fungi and mycology. But I would recommend getting a foundational text like that. Texts like, texts like Paul Stament's Growing Gourmet Medicinal Mushrooms, or David Aurora's Fungi Demystified, or even just a good field guide in general. Those all have their own aspects, but there's very much either the focus of finding mushrooms for IDing them, learning about their ecological roles, finding them for the culinary value, or even finding them for the aspect of culturing and then, taming that wild fungus in an aspect, or even getting it to the point where you can then cultivate it on your own property in an emulation of its natural environment.
So if someone wants to get hands-on with it, what's a good first introductory project for them to get started with?
I think like just the biggest thing that I do is for teaching is that I teach a lot of, just working with fungi. So we're very lucky in a time where we have a number of people that produce mushroom spawn, like the mycelium growing out on a substrate. And what I'll do is I'm very lucky very lucky to be good friends with Smallholder, a mushroom farm in New York City. And they produce a sizable amount of spawn in their production. So I'll pick up some spawn, either a spent block, quote unquote spent because it's still biologically active, or a fresh block. And I'll just teach people how to run that mycelium on cardboard. So often working like a species that is a white rot fungus, the gourmet mushroom, mostly your white rot fungi, meaning they're more of the tenacious decomposers, like an oyster species. And what we'll literally do is we'll just get a basin, like a tote. We'll get some water, preferably spring water. Oysters are pretty tenacious; they're not that delicate. More so because of chlorine and additives to water that may interfere with microbial diversity, but oysters are pretty tenacious. And we'll just pasteurize. We'll boil the water, we'll pasteurize the cardboard, we'll hydrate the cardboard, but we'll also heat it up to kill any competitor organisms. And then we'll strain it off, and then we'll make little, mycelium burritos. So taking some of the spawn and inoculating the cardboard and letting people understand that this organism, they can watch it grow and run and consume the cardboard and get the experience of working with mycelium in a way that's like to the kin of like planting a seedling and watching it basically sprout from the seed and watching it grow. And then learning that, fungi being kind of amazing, they're like this holographic organism, they'll continue expanding. Like the fact you can have a culture of mycelial tissue the size of your pinky, and then you could expand that out to, for a whole full-scale mushroom operation. There's truly a fact of this organism, if with the right application, can continue to expand and grow. And then even dropping that thing to them, like on one end, like, okay, here we are with a single block of mushroom spawn, 5 to 15 pounds. But then telling people, given the right conditions and natural ecologies, we'll tell them about the humongous fungus, like this mat of this 3.9 mile mat of mycelium of honey mushrooms, Emerlia ostiae in the Cascades. That's somewhere in the range of like 40,000 pounds. And by DNA work, they've confirmed it's one contiguous organism, which blows people's mind. Like the humongous fungus is the largest organism on this planet.
There's so many facts about this organism that just blow my mind like that.
Yeah, so that's the introduction, the fact that like it's something that it's hands-on, they can get it. And oftentimes a lot of teaching, the more cerebral you get, depending upon that person's learning style and their attention span and the tone, and I kind of teach in a very animated tone where I'll maybe go in depth, but I'll bring it back to like a fun fact thing. But just the hands-on application of just getting some mushroom spawn, inoculating some cardboard, and learning how to do it successfully, but also learning how to do it wrong. Like learning from the mistakes, because you can observe what you did, and you can actually do that little bit of science and understanding like what are the variables of my experimentation in a very rough form, without being kind of learning about the scientific method and ways to record data in a more meticulous academic matter that may be intimidating to people who just want to explore and learn and get it to learn on their own kind of feeling and learning pace.
I see that as you helping them get that initial connection to the information and then allowing it to sort of grow out from there, and they can go as deep or not as they want. Now, are you doing this on your own, or are you affiliated with an organization?
So that's a great question. So I started It's kind of funny. So I started NYC back in 2018, and it was kind of interesting because I was just me doing a project where I wanted to just study micromediation, like, and it's something where it's literally just you running, my like tissue samples of different white rot fungi, the select remediated candidates on agar, and kind of observing their growth rates and introducing different substrates. It's very boring work. It's not interesting, or well, maybe if you find something interesting and share it, but it was just me doing that, and so I was interested. And so I got to start at community labs back in 2018, and so I was just initially just me doing it, and I was kind of like, so this was, I thought I was gonna be just me doing it in a lab, and they asked me, hey, this is a cool project, would you want to open up to the community? And I'm all about open science and collaboration, and I'm like, okay, cool. Maybe like 5 people will hop on, 3 will follow up, me and 2 people kind of working on it. And then I was thrust in the scenario where there were 10, 15, 20 people showing up to these sessions. And I was like being thrown into this teaching scenario. And it was kind of very kind of feet first situation of like getting into teaching and learning as well, but also like asking myself, how well do I understand something? Because learning something to do by yourself is one thing, but learning how to do something to teach other
Teaching
people.
is a great way to learn.
Yeah, it is. It
You have to
is.
know it.
So kind of that's how I got started. And then it started out with just a name from an Instagram page. And then eventually, this continued on and create opportunities where I could teach and get paid for it, and kind of started as a DBA, kind of a doing business as for myself. And I kind of evolved from that. And, so yeah, NYC it's an educational resource based in New York City that teaches the benefits that fungi provide to our environment and our health and our society.
And you're
And
holding like paid classes where people can pay money and attend one of your sessions and get some hands-on
Yep.
practical experience.
Yeah, I was doing that. I was doing paid and I was public and private classes. So public classes were usually through the community biology spaces, of which there are 2 in New York City: Gen Space, Biotech Without Borders. Both of them were founded by Dr. Ellen Jorgensen, who's an incredible mentor. She's actually and so it's interesting because she's all about open science, and mycology is new, interesting thing, and also a number of interesting things with molecular biology with fungi. So I got my start there, and then that moved on to being invited to being asked to speak at different universities or colleges about fungi in different classes, whether it's just kind of microbial ecologies, just general science classes, even biodesign classes, talk about like bioart and all different ways. And I had a very interesting range of experiences and kind of filled this really interesting niche where I'm like, okay, this is really interesting. I'm like doing science, but I'm also engaging in more practical ways as well. So yeah, so it's something that really at a certain point it started as just an educational resource, and kind of became like this this name I wanted to like be eponymous with mycology in New York City. Whereas kind of it's also an iteration of myself as kind of the sole person that runs this thing.
That's
So
pretty
yeah.
cool. You kind of just kind of fell into it, which is often the best way for things to happen. You didn't force it; it just happened naturally.
Yeah, and it's kind of funny. It's kind of like much like the metaphor of a mycelial network, which is not only decentralized but distributed. That when you're getting involved in this and you connect with people, you find these other distant networks and nodes that are doing similar things, and it's kind of nutty. Nutty in the best way, crazy. I have friends that are all over the country that I talk with over social media or text about similar things, in a whole range of walks of lives and careers, but all kind of bound together by, being mushroomed, like about, with this passion, with, also they're very beautiful reproductive forms of fruiting bodies, mushrooms, but also the fact that fungi are just kind of elusive and mysterious and have all this potential, and what we can learn from them. Yeah. And so not so much what we can learn from them, but what maybe they can teach us, and they're more subtle, more pervasive and ubiquitous roles they have in nature.
There is something special about this organism in its ability to connect people together. I've found that just with running the podcast and doing EverymanBio. There's a sort of kinship that happens amongst people that are bitten by the bug of studying fungi. Something you touched on that I think is interesting is I think there is a growing movement of people who are exploring mushroom related businesses. You sort of have a business now that you use that's founded on teaching mushroom related information and proliferating that out to interested people. There's other people that are interested in farming mushrooms and selling them, and there's people that do sell cultures. Maybe you could talk a little bit about that. What opportunities are there for people who might be looking at creating a business around mushrooms?
Yeah, and it's something that it's really unique because I think in general in people that are interested in, being their own boss or working for themselves, or like, starting a company or a business or what have you, it's something that's unique because we're still in the early phases of it, right? Of people actually doing it. Like, I think there is a bit more public awareness of fungi, and they're starting to pop up more in pop culture, and even in designs, and it has kind of entered the subconscious of kind of mass media and pop culture. But with people like who are cultivators of mushrooms, whether they're curators of cultures, whether they're people who are even just making art, like it's kind of amazing, like this whole aspect about how fungi have become popular and it's an actual viability, and even too, like people that work with chefs who love the expression and flavor of profiles. So there's all these opportunities that that are there in fungi, and this kind of dedication and passion. Whether they're people that are even producing supplies for cultivators, producing like producing agar plates and substrate for people who may not have the space. Whether people are just learning to catch the bug, where they want to experiment as a hobby or turn it into a business. So I think there is quite an interesting aspect of where it's a very unconventional path, and where I think a lot of times, like especially in for Americans, and like people maybe in their Generation X, millennials, and Generation Z, a lot of the narrative is like, oh, go to college, get this, do this as well. And, unless you had a good time to think about what you want to do, you're kind of pushed into this track of higher education, which you're learning things, but, the translation into a career and viability, where people are realizing like they can become passionate, but also generate an appreciable income that can, provide for themselves, their families, and even their communities. So yeah, I think it's pretty amazing, just the whole range of potential for careers and even businesses and even different entities that can exist in. So it's pretty amazing how, because they're so new and in a larger scale thing, and really what I see now is like really where you think, oh, competition, but the reality is that there is so much opportunity and so much demand that is yet to grow. There is so much potential for people to start businesses, and even something I've noticed in kind of the communities, there's far more willingness for collaboration than competition, right? There's a bit of competition, but the reality is we're realizing that it's kind of breaking down the narrative of like, like the survival of the fittest, every man for
There's
himself.
a network effect that gets created, especially with early
Exactly.
stage industries like this industry. Everybody helps each other rise. Absolutely. What do you think is the cutting edge of mushroom research right now, and what do you think we're going to see in the next 5 to 10 years in terms of research and innovation and perhaps breakthroughs in the space?
Yeah, so it's really, it's kind of crazy thinking about the whole broadness of it. People now are really kind of diving into understanding fungi as medicine through their potent immunomodulatory effects, and they're getting more and more attention. And it's pretty amazing because a lot of the history, a lot of the research, more so coming out of East Asia, has been around the fact that like most research understands like this whole range of compounds interacting together versus a reductionist single compound that often is based around a kind of a means for patent or a profit, because you can categorize it and closely study it. So it's kind of interesting because the whole range of the factor, I think some of the most exciting things is the molecular mycology. The fact we can understand some of the amazing genetic diversity these fungi have to do different certain applications. Because it's pretty amazing how, this is something I always think about now, like I think about, especially when culturing fungi, how you're finding a fungi out in the wild, and they're anything but fragile. They're living in this environment where they're in constant breaking down resources. They're fighting off pathogens, whether they're bacterial, viral, or even fungal, and producing metabolites that persist with it, but even collaborating with other organisms and they're expressing all these interesting ranges of genes that are either stimulated by their environment, stimulated by themselves, or stimulated by competitors, pathogens, or even collaborators. So I think this is something too, is like definitely in synthetic biology, one organization that I ran a team. I helped run a team last year, instructed a team, and even this year I'm mentoring, is iGEM, the international iGEM competition, which is using molecular and synthetic biology to kind of create solution for real problems. And synthetic biology is exciting because we're looking at systems now where we can harvest the genetic diversity, synthesize these genes, and produce circuits that can even function in cell-free systems. So one of the most exciting things that I'm looking interested in, like that's a practical system right now, is the model for where this could go, is the ability for biosensors. The ability where, I think about in a lot of environmental science, how, in the environment there's organic contaminants, there's inorganic contaminants, there's contaminants that are stable in one form, but based upon the time of year can be changed, become toxic again. But oftentimes the way you assess these is by taking a sample, and you're doing an chemical analytical process that is often expensive, takes a lot of time, and the results are hard to interpret, and it's weeks later. Whereas now you literally utilize the fact that organisms are interacting with these contaminants in the environment, and take a sample from the base of the soil, pipette it into a test tube with this organism that has a genetic circuit where it has it has a gene for a enzyme or protein that binds to a heavy metal, and then further down the circuit you have a gene that's going to express fluorescence or a chromopigment protein, and within a matter of minutes, not weeks or months, you can get relative results that can inform a decision. So I think with synthetic biology, being able to fact that the cost of synthesizing and sequencing DNA is getting cheaper and cheaper and cheaper, that liberates the factor. There's an amazing graph by the NIH, which looks at the cost of DNA sequencing per megabase and cost of DNA per genome. And you see this graph where it's like Moore's law, this nice tight 45-degree angle roughly, where every 6 to 18 months the number of transistors on a circuit would double, and the price would either basically reduce by half in that time frame. Whereas Moore's law, this nice tight 45-degree angle, where the cost of sequencing DNA just went like that to the floor. And it's kind of mind blowing because the biology revolution, the revolution in molecular and synthetic biology, is happening even faster than the computational or the information revolution.
That's fascinating.
This is something that I think there's a huge potential, and a number of organizations like iGEM are trying to teach the understanding of bioethics, or biosafety, and understanding that like we have a great responsibility because we have this power of understanding what are the implications of how these things work, but also how they can be used to function for good as well. So that's the exciting thing I see, is the applications for synthetic biology in the future. But with mycology, definitely people learning how to cultivate species that are a bit more selective, and even getting better at scaling these things for incorporating fungi into waste streams, really closing the loop and developing these truly regenerative systems. Like I would love to see one day where we understand the relationships between fungi, bacteria, and other organisms soiled, where you would, rather than having a dump, you would have your community bioreactor.
Absolutely. That's my dream as well. Well, hey man, what's the best way if someone wants to reach out to you and connect with you? What's the best way for people to find you on the web?
Definitely. So best way to find me, you can email me, and I'm happy to send this over. You can put it in the show notes. Craig, C-R-A-I-G, at MYC, that's M as in Mary, Y as in Yankee, C as in Charlie, .NYC, N as in Nancy, Y as in Yankee, C as in Charlie. I thought it was a cool moniker at the name. I realized it was a bit of a synonymous palindrome nightmare when I'm trying to tell people, but it's MYC, NYC. Instagram is at nyc.nyc, but if you just go to the domain myc.nyc, it'll forward straight to my Instagram page. That's the best way to reach me as well. So so I don't really post that much on my story, but When I do have classes or events, I don't post that much on my timeline, my feed, but I do post regularly on my story. I'm working on a number of projects this fall, which are more educationally related. I've been accepted into Cold Spring Harbor Laboratories, their DNA Learning Center. They have a urban barcode research project, so teaching high schoolers molecular biology and DNA barcoding.
Wow.
I'm also planning to work with Sigrid as well in that junction. She's been elevated as kind of one of the board members for Fundis, the Fungal Diversity Survey, and
Wow.
even thinking about putting out a handbook, using a handbook for people that are interested in getting into molecular biology through fungi, because there's a lot of like information that's all around and it's not consolidated. So, but yeah, so to answer, yeah, the best way to get in contact, or either through my email address or Instagram. That's the best way to reach me in general.
That's amazing. If there's anything I can help with those projects, even beta test something, feel free to run it by. I'm
Yeah,
happy to help any way that I can.
definitely. There are a lot of online collaborative platforms like protocols.io or Joggle, just one giant lab, where there are measures where you can create a network that's not hosted on any like platform like Facebook that has an incentive to serve you ads or to constrain what you see through an algorithm, that can really do open science. And I think I think there's a lot of cool projects, definitely working with people who are, either have a background in molecular biology or learning it. Because I think a lot of this aspect, there's a lot that can be done to progress mycology into the molecular age, and how much we can learn from an ecological standpoint, from a science standpoint, from, an applied standpoint. So that's kind of the exciting thing.
Absolutely, man.
Yeah.
Hey, thank you so much for joining. This is truly an honor, and I hope we can have you on again, because you're just a wealth of knowledge.
There are definitely a couple of things we wanted to talk about. A lot of cool projects for sure. So I'd be happy to be like a guest on as well.
Yeah.
But yeah, definitely, Josh. Thanks for having me on. Keep up the good work. Keep pushing.
Thanks, man.
Definitely, I'm loving the view of the laboratory you got on. Since COVID, my lab space kind of got shuttered, but I've been focusing more on like diving into the papers that I picked up for the past year.
It's a good time to do that. It's always a good time to do that. So
definitely, for sure. All right, Josh. Thank you.
all right, man. See ya. Cheers.